Hello, everyone. In this chapter, we are going to cover complex traits. So, what are the learning outcomes? We are going to distinguish between single gene and polygenic traits. We will define what complex traits are. We will explain how continuously varying traits reflect genetic and environmental influences. We will explain how determining empiric risks differ from calculating a Mendelian frequency. We will define the concept of heritability, and then we will discuss for adopted individual and twins how genes affect their appearance and also how the environment affect their appearance. And then we are going to dive into some methods that researchers use to study the variation or risk of occurrence in a population, a particular approach that is called genome-wide association study or GWAS. And then we are going to discuss how researchers apply this method to investigate a number of features in a population. So our first step is going to be defining complex trait. Now, as you may remember by now, only in few cases, very limited cases, one single gene is responsible and can control a phenotype. Very limited number of cases, right? So, one gene inheritance is really maybe the exception, right? Most of the time, you are going to have many different aspects that are involved in someone phenotype. So environmental factors and other genes are going to be involved in how genes are being expressed. Now, there are two concepts that I want to draw your attention to. One concept is the Mendelian concept. In the Mendelian concept, we call Mendelian trait a trait that is caused by one single gene. When a trait is controlled by more than one gene, then we are going to call that trait polygenic, right? Multiple genes. But I want you to remember as a take-home message that there is no one feature that is ever controlled by one single gene. In fact, we use the term multifactorial to underline the fact that many traits that occur in a human are actually caused by multiple factors, including genetic background and the environment, right? Many studies show how much genetic background and the environment can play a role in someone's appearance. For instance, the skin color, the hair color, the hair shape, right? Many things actually play a role in the traits that we can see. Nevertheless, when it comes to human genetics, we want to know what is the proportion of genes that explain a particular phenotype. What is the proportion of the environment that explains a particular phenotype. So, needless to say, that it is going to be complex to try to isolate the role of a particular gene or a particular set of genes in the physical appearance that we see in individual. This physical appearance can be something that we can see with our naked eye, something we can touch, but it can also be very much biochemical, right? Which means the reactions that are happening in our system. I want you to keep that overall observation in mind as we cover this chapter. Now, when we consider, for instance, genes that are involved in causing lung cancer. Now, lung cancer, it's actually a trait that is very complex because you have a number of variants of genes that increase the risk of becoming addicted to nicotine and therefore to developing lung cancer. Nevertheless, in spite of these genes that can predispose someone to developing lung cancer, that doesn't mean that an individual will develop lung cancer. Now remember that smoking, it's a lifestyle. It's a choice that one's make, right? So if one decides to not smoke, you can very well have the predisposition, genetic predisposition for developing lung cancer and never ever develop lung cancer. So please keep that in mind. So having traits that are purely polygenic is actually extremely rare for the reason that lifestyles plays a very important role. The environment plays a very important role. That's why there is hardly any situation where multiple genes are exclusively going to explain a trait. Now, polygenic traits are very complex. If you consider, for instance, the height in a population, the skin color, the body weight, and many other illnesses in a population are going to be complex traits. And we are going to throw in the word complex to actually underline the fact that not only genes are responsible for that trait, the environment plays a role, what you eat plays a role, what you get exposed to plays a role. So the polygenic complex trait or the condition are basically the consequences of a contribution of several genes. And again, remember, not only genes are involved, but the environment is also involved. Now, here is the thing though. Each gene is going to confer a degree of susceptibility, but the level of contribution of all genes is not necessarily equal, which means that even though we may have similar genotypes, we can actually express the trait in a different manner for virtue of the degree of susceptibility or the degree of expression of each of the gene. So different genes may contribute different parts of a phenotype. For instance, in a case of migraine, there are a number of genes that are going to be involved, and not only they are involved in different manner. That's a very important detail to remember. For instance, you have a gene, in a case of migraine, you have a gene that is carried by chromosome number 1. That gene that is carried by chromosome number 1 plays a role in the sound, susceptibility to sound when you are developing migraine. On the other hand, there is another gene that is carried by chromosome number 5, and that gene is involved in the pulsating headaches and light sensitivity. So migraine that involves sensitivity to sound, sensitivity to light, and headaches, now you see that separate genes on separate chromosomes are going to affect these different aspects. To make things even more complicated, when one has migraine, one experiences nausea and sometimes vomiting. Not systematically, but sometimes vomiting. Now, nausea and vomiting are associated with a gene that is carried on chromosome 8. Now, I want you to imagine the complexity of isolating the role of each of the gene when it comes to characterizing the genetic background of migraine, right? This, you can anticipate the level of complexity. And that is the very reason why you are going to see there are a lot of concepts that are part of this chapter that are rather complex, right? So bear with me and listen carefully. And if there is anything that is not clear, write it down and reach out to me. Now, polygenic traits are going to be continuous. You have a continuous variation in the phenotype. But maybe before I dive into that, let me digress and go back and saying that in the case of single gene traits, the case of single gene traits are going to be discrete or qualitative. So the question is that trait being expressed yes or no, right? So we have an all or none effect for single gene traits. Now, really, we are exaggerating. I am exaggerating when we say it's an all or none effect. But we know things are more complex than that. But remember that single gene traits are discrete because you can isolate the single gene, right? They are also called qualitative traits. While the polygenic traits, because there is a variation of phenotype among people and there is no limitation as to how much variation is happening for a given phenotype, you are going to see a huge array of the same thing. That huge array of the same thing that is characteristic of polygenic trait is called quantitative threat. That quantitative threat brings another concept. It's the concept of quantitative threat loci, or QTL. QTL, it's basically genes contributing to polygenic threat, right? Genes contributing to polygenic threat. If we go back to the case of migraine, we are going to ask the gene that is carried by chromosome 1, what is its contribution to migraine? The gene that is carried by chromosome 5, what is its contribution to migraine? At which case, these genes are contributing to the severity of migraines, because let me remind you of the fact, well, I suffered of migraine, right? And I know many other people who suffer of migraine. Now, I've grown out of my migraine, so I don't develop migraines anymore. But the level of severity of migraine that I have was different from that some of my friends had, right? So this may account for the variations in the genes. Say my gene of chromosome number one can be different from that of my friend, and that is going to account for the variation that we see in the phenotype, which brings us to the quantitative threat loci. One locus, several loci. Okay, so the frequency of distribution of phenotype is going to typically form a bell-shaped curve. That's what we see with polygenic traits. Regardless of the polygenic trait, we are going to see a bell curve just like this. Now, the fingerprinting patterns, it's a good example of this. So the skin on the fingerprints, it's typically folded. When you look at your thumb, your hand, you are going to see some ridges on your hand, right? These ridges are caused by some foldings that happen within the dermis. Now, your skin is made up of two parts. You have the epidermis and the dermis, right? The dermis is made up of irregular, condensed, loose connective tissue. Underneath of the epidermis, you have the dermis. And that dermis is going to be made up of a huge amount of connective tissue. And that connective tissue in particular is the dense, irregular connective tissue. That dense, irregular connective tissue is basically that the way the protein fibers are aligned, it's not following a pattern that is regular, causing the dermis to be folded in a way that form ridges in the hand, right? So those ridges are going to align in a certain way, form loops, whorls, and arches, and all of that is going to lead to patterns that are going to be the finger printings. And these finger printings are going to vary from one person to another. There are not two people in this world that have the same patterns of fingerprinting. Even true true twins, even true twins that have the same genetic background, right? Because of the rubbing of the hands against the walls of the womb, well, this is going to cause a different pattern among monozygotic twins, right? Now, there is a whole discipline that studies fingerprinting. That discipline is dermatoglyphics. Dermatoglyphics studies fingerprinting, and this is when, we can tell people apart. For instance, when tracking, for instance, a crime, we can tell people apart. Now, obviously, these days, technology has evolved in such a way that, everything is done by a computer. So, the number of ridges is largely determined by genes. But not only that, but also prenatal contacts. I was telling you about the hands rubbing against the walls of the womb, right? Now, with that said though, the average total ridge count, it's going to be about 145 in males, while that average is 126 in females. So there was a study that was done among students to actually look into the total ridges from a point A to a point B, so from here to here, among students. And because this is a polygenic trait, right? We said that the polygenic trait is going to describe a bell curve. Take a look at this. So we have here basically a bell curve with an average that is going to be, for most people, are going to have a number of ridges, right, that are about 170, right, to look at what we have here on this graph. However, you have a limited number of people who are going to have a very low number of ridges that are below 100, while you have also a very limited number of people who are going to have number of ridges that are above 200, right? So that's basically how you should read this graph. You have an average that is going to be within this area where you're going to have the most people with a number of ridges that fall between, say, 130 and 190. All right, how about the height? The height, it's a combination of effect. Combination of effect that involves both genetics and environment. Now, what someone eats plays a very important role in the height of an individual, right? If someone experienced malnutrition in a very early age, chance that they're going to grow tall is low, just because they were malnourished, right? In the United States, for instance, the diet is so rich in nutrients that overall children are growing much faster, much taller here in the United States than in many parts of the world, right? And very importantly, people from the many past decades, overall, have had a height that were lower than people today, because today people overall are in better health. They are eating food with better nutritional values. Because of what we know about food today, we can specifically select the food based on the nutritional values. While if you go many decades back or centuries back, well, it was not about the nutritional value, it was about just getting fed, right? So I just want to be fed. Now, think a minute what happened in Europe many, many years ago, right? During, for instance, the period of Great Depression, okay? People didn't have much to eat. They ate whatever was available, right? As consequence of that, well, these people got affected as to what their height was. So anyway, to come back to the genetic portion, you have a bunch of genes, at least 50 genes are involved in the variation of height. Some genes are going to have a large effect as to their contribution to the height, to the variation of height. Some are going to contribute to a lesser effect, right? So we can see here in this photograph, we're going to see a bell curve in terms of the height, right? You're going to have a limited number that are very short, a limited number that are very tall, and the largest number of people is going to be within a certain range of height with a particular height here that is where we have the most number of people, right? The skin color obeys the exact same type of pattern, right? Melanin that is produced in the skin is going to protect the skin against DNA damage from the UV, right? So exposure to sun is going to increase the synthesis of melanin. So the more exposed we are to sun, the darker our skin is going to be. And guess what? That doesn't depend on how many genes are involved in the skin color. Just because we are exposed to the UV, a natural response of our body is going to be high production of melanin, right? For the sake of protecting our cells against DNA damaging effect of UV light. So we know humans across have the same number of melanocytes, but the number of melanosomes and the size of melanosomes, the density of distribution, is going to change from one person to another based on where you live, based on your lifestyle, based on, well, also your genetic background, right? More than 100 genes affect pigmentation in the skin, the hair, and the irises. So here you have a bell curve of the skin color. Notice the presence of dominant alleles and recessive alleles, right? The more recessive alleles one has, The lighter the color, the more dominant allele one has, the darker the color. But we are going to see that the more the combinations of alleles is really what is going to determine the shade of color skin. And we see that this shade of color skin, it's actually continuous, just like the height was a continually variant trait. Now, here we have two kids that obviously color is not the same, right? And because of that, humans have decided that these two kids whose color is not the same are from different races, right? We're going to say that one is white, one is black. But really, the concept of race doesn't exist biologically. There is no concept of race at a biological level. This is a complete social construct, right? Regardless of how we want to put this. Okay? So the definition of race based on color, it's completely a social construct, not a genetic construct per se. Because when we look at the color of skin based on genetics, we realize that, well, we cannot predict the color of skin based on genes. Because 93% of varying inherited traits are no more common in people of one skin color than any other. Okay? So, really, we have to be cautious with the biases that social construct are bringing when it comes to genetics. And actually, a lot of the ideas of the social construct influenced a lot of research, and particularly in the United States and Europe, as to genetics, because people try to explain some racial differences. When I say racial differences, I mean the social construct, right, of racial differences by genetics. But we know today that, well, it doesn't equate, right? All right. What techniques do we use to study complex traits? Well, we are going to use a number of techniques. One is, for instance, the empirical approach. In the empirical approach, we are going to use the concept of empiric risk. The empiric risk actually measures the likelihood that a trait will occur in a population based on incidence. Based on incidence. So one is looking at a population, right? We look at the occurrence or the recurrence, how often this is happening repeatedly in a population, right? So that we're going to focus our approach by observation, right? Which is going to lead to another concept, that is the incidence. The incidence is the rate at which a certain event occurs in a population. For instance, how often do we see cystic fibrosis in a population? Now, obviously, in the case of cystic fibrosis, we said it was a Mendelian trait, right? So that may not be a good example, okay? Migraine would be a good example, right? How often do we see migraine, people with migraine in a population? And then another concept is going to be the prevalence, which is what is the proportion or the number of individuals who have a particular trait at a specific time. So, empirical risk is going to be we observe, right? What do we observe? That's beyond theoretical, right? What do we observe? Two, incidence. Incidence, what is the rate at which a certain event is occurring? Three, prevalence. What is the proportion? What is the number of individuals who are affected by a certain threat at a very specific time? Because prevalence can vary over time in a population. All right, so empirical risk is not a calculation, but empirical risk is based on population statistics, based on what is observed in that population, right? So, which means that everything is going to depend on really the size of the population we are looking at, right? So the population might be broad, such as an ethnic group or a community, or a population can be more well genetically defined, right? Such as a family or families that have cystic fibrosis, right? Now, empirical risks are going to increase with a number of factors. For instance, the severity of the disease, the number of affected family members, and how closely related people who are affected are, right? How closely related people are. So empirical risk is going to be involving severity of the disease, number of affected people in a family, and how closely these people are related, right? These are some aspects to take into account when looking at empirical risk. Now, an example is the investigation on the occurrence of the cleft lip, right? The cleft lip right here, okay? The cleft lip is more likely in a person who has relative with that condition, right? If there is no one in the family, close or extended, that ever had a cleft lip, this can only happen because of environmental issue, because one single mutation cannot cause that to happen. It has to be a broad issue. For instance, a teratogen, right? And remember, a teratogen, it's an environmental factor that is affecting early development of an embryo, right? That can affect, at the end of the day, the development of some organs. All right. So when looking at empiric risk, for that particular feature, the cleft lip, For identical twin, that risk is about 40%. For siblings in the same family, that risk fall drastically to 4%. That's 10 times less, right? For a child, it falls to 3.5%. When we say for a child in relation to parent, right? Niece and nephews, it falls to 0.8%. First cousin, 0.3%. In the general population risk, no affected relative, 0.01%. So really, the empiric risk, it's going to be as close you are together, the most likely the chance, right, to develop that feature. How about heritability? Now, this concept, it's a little complex, so please bear with me. Heritability. Heritability, it's actually represented by the letter H. Heritability, it's an estimation of the proportion of variation in a complex trait due to genetics in a particular population at a very specific time. Okay? So, heritability, it's a way to estimate, right, the proportion of variation of a complex trait, right, due to genetics in a particular population in a particular time. Heritability is the degree to which variations of a trait are occurring due to genetics and not to the proportion of the trait itself attributed to the genes, right? So now I know it's a little bit complicated, all of that. So basically, we are not focusing on one particular gene. We are focusing on the genomics, if you will. We are focusing on the genomics. What are all the genes that contribute to a particular trait? That's one. Two, what is the degree to which we see a variation because of that genetics, right? So that we are going to come up with numbers. Heritability that equals 1, it's a trait whose variability is completely the result of gene action. Let me repeat that. Heritability equals 1 for a trait for which variability is completely the result of the action of a gene, right? However, the variability of most traits are due to differences among genes and the environment. Let me repeat that. The variability, most of the time, for a given trait, is going to be the combination of not the combination, but the difference, right? The difference among genes and the environment. Okay? So that's heritability. Now, when considering heritability of complex trait, we have to take a look at a number of aspects. We have genetic variance among individuals. We have environmental variance for the space in which individuals are living, right? For the genetic variance, we are going to see that most of the time, we're going to see an additive effect of recessive alleles, while we have a limited contribution of dominant alleles, and also we have epistasy. Now, epistasy, it's an affection of gene expression, but not gene themselves. Let me repeat that. Epistasy is an affection of gene expression, but not the gene themselves, because there is nothing wrong with the gene. But there is everything wrong with gene expression, because, for instance, of the addition of a methyl group on a gene, right? So, heritability is going to be a number. It's a number, right? And that number, to get that number, you have to take into account genetic variance and environmental variance. All right. Heritability is going to change as the environment changes, right? Heritability for skin color, for instance, is going to be higher in winter months when sun exposure is less. Now, sun exposure being less, we're going to have less melanin synthesis, right? Okay? And less melanin synthesis is going to cause, heritability in the winter time to be high, right? If you take children born from Caucasians, chance that being born in the winter time and being exposed in the winter time when light from the sun is very low is going to cause individuals to have lighter skin overall. While when time changes, in the summertime, the skin is going to become darker for the same individuals. All right. So, another thing is that in population from Equatorial Africa, like myself, I come from Gabon. Gabon is a country that is crossed by the equator line. So population in Equatorial Africa are going to have darker skin than people who come from the Scandinavians area. Okay? In the Scandinavian area, there is hardly any sun. Therefore, there is not much UV to be protected against. So no much melanin is produced, right? And then you have a concept which is the missing heritability. The missing heritability occurs when identified genes cannot account for all the variability in a trait or a particular illness. Let me repeat that. Missing heritability occurs when some very specifically identified genes cannot account for all of the variability in a trait. Now here we have heritability for some human trait, for instance, the club foot, okay? Heritability is 0.8. Remember, complete heritability is 1, right? So the lower the heritability, the lower the contribution, right? So then we have some other heritability. For instance, the body mass index, it varies between 0.4 to 0.7, right? Now, I'm not going to ask you to remember this irritabilities. I'm not going to remember to ask you to remember that. What I want you to remember is the concept, right, of irritability. All right. So we can calculate irritability and we can estimate irritability, right? Typically, researchers are going to use different statistical methods to estimate irritability. One way, for instance, is to compare the proportion of people sharing a trait to the proportion of people who are predicted to share that trait. Let me repeat that. Proportion of people who are actually sharing the trait to the proportion of predicted people to share that trait. That expected proportion is derived by knowing the relationship that exists between individuals, which means that using a pedigree can be a very great source of information to estimating heritability, right? Tracking the relationship, the blood relationship among individuals. So then, another concept is the coefficient of relatedness. What is the coefficient of relatedness between individuals? Now, if you consider siblings, so siblings to sibling, the coefficient of relatedness is going to be 50%. Why? Because siblings have the same parents, right? They have the same parent. They receive 50% from mom, 50% from dad. So when you look at siblings to siblings, the number of genes shared or the coefficient of relatedness, it's 50%. However, when we look at the degree of relationship, that degree of relationship is 1 between siblings to sibling, right? Parent to child, that degree of relationship is 1. Uncle and aunts, aunt to nieces and nephews, it's 2 degree. Okay? Now, notice that the coefficient of relatedness is going to be 25% for nieces and uncles, nephews and uncles. Half siblings, their degree of relationship is 2. Okay? The coefficient of relatedness is 25%. And all of this is very logic and simple, if you will. But if something here is confusing, please let me know. Now, so we can look at the degree to which people are related, right, through this pedigree. So if we consider Tim right here, okay, so Tim has a degree of relationship that is 1 with Tina, because obviously there are siblings, right? And to Tom, that is 1. There are siblings, right? But if you consider, for instance, Tim in relation to Olivia, okay, they have a degree of relatedness of 2, right? Because Tim to Olivia is the uncle of Olivia. Now, if we go back here, uncle to niece, it's 2. All right? Now, adopted individuals are going to share little with their adopted parent when it comes to genetic makeup. However, they are going to share a lot with adopted parent when it comes to environment, right? So the similarities that exist between adopted people and adopted parent is mostly the consequence of environmental factors, while the similarities that exist between adopted children and their biological parents is going to reflect mainly genetic makeup, right? Now, the information in both, well, actually, I should say the three, the adopted people, adopting families, and biological families are going to basically allow to connect how different components are playing a role in the threats that we see, the role of the environment, the role of genetics in the threats that we see. For twins, the study on twins allowed to come up with another concept. That concept is basically concordance. Concordance is basically the measure of the frequency of expression of a trait in both members of a monozygotic or a dizygotic twin. So, in other words, and in a simple way, if two twins are different in a particular trait, we are going to say that they are discordant. They are discordant, right? Because they are different, right? So the more influence gene exert over a trait, the higher the difference in concordance between monozygotic and dizygotic twins. Makes sense? Because the monozygotic twins, they are perfectly genetically identical, right? While the dizygotic twins are going to be very different genetically, right? So, if the genes play a very important role if a gene plays a very important role in a particular trait, the monozygotic and the dizygotic twins are going to have a huge difference in concordance. In other words, they're going to have they're going to have discordance, right? All right. So here we have on this slide some features that were studied for concordance. I'm not going to bug you with these values. I want to bring these values just to your attention so you can kind of see how monozygotic twins and dizygotic twins relate when it comes to the concept of concordance. For instance, acne, the occurrence of acne in monozygotic twins and dizygotic twin, it's exactly the same. Okay? While for Alzheimer's disease, it's very different, right? So for monozygotic twin, in other words, if one has Alzheimer's disease, chance that the other one is going to have Alzheimer's disease is very high. It's very predictable, right? But in dizygotic twins, when one has Alzheimer's disease, that doesn't mean that the other one is going to have Alzheimer's disease. Okay? So you can kind of see. This is just to give you you know ways to appreciate this. If you consider, for instance, autism, the concordance value in a monozygotic twin, it's 90%. So very high, right? However, between dizygotic twins, it's 4.5%, very low. Now, between twins, the taste, the preference, and the choice of a certain number of things can be actually very shared among monozygotic twins more than they are in a dizygotic twin, but it can also very much vary. But we see actually a concordance in preference, in taste, just like what you see here on the picture. Now, what can we do to try to separate the proportion of genetic and the environment for a given trait? Let me tell you up front, this is difficult, right? It is difficult, okay? But to try to achieve this, researchers have shifted interest on monozygotic twins and dizygotic twins. So dizygotic twins share an environment and 50% of genes. Monozygotic twins, they are identical genetically and share an environment, right, when they are developing. Twins raised apart, they are going to share genotypes, but not the environment. Adopted individuals are going to share the environment, not genes. By selectively working in these different groups helps geneticists to actually be able to find leverages to separate the role of genetics and the role of environment as to the traits that are being studied. Now we are going to look into the genome-wide association studied, or GWAS. The GWAS actually compares many genetic markers across the genome between two large groups of populations. For instance, a population in Tuscarawas County compared to a population in, Holmes County, right? That would be an example. So we are going to take a look at this population based on traits that exist in one population and that does not exist in another population. So it's going to be a comparative study, right? Now, using the genome-wide association study, we are going to be able to identify DNA sequences that are much more common in affected people in one group, right, compared to another group. What is the contribution of the suspected genes in one phenotype compared to another group, right? So there are two major approaches to the GWAS study, right? Genome-wide association study. We have the single nucleotide polymorphism, also abbreviated SNPs. The second one is the copy number variant, CNVs. The SNPs and CNVs have been used to actually do a genome-wide study to actually look at the occurrence of some diseases, some features among peoples. SNPs are actually going to focus on looking at a single nucleotide polymorphism. In other words, one single nucleotide in a DNA is varying in a population, right, at a particular site of the genome. And this is when this is happening in more than 1% of the population. When this change is happening in more than 1% of the population, we are going to say that, okay, yes, there is a single nucleotide polymorphism in a population. If it's in less than 1%, it doesn't count, right? The CNVs, it's basically a repeat of a particular sequence. That particular sequence can be CGTA, CGTA, CGTA. They have to be organized in tandem. Tandem means one comes after another, that comes after another, that comes after another. That's a tandem. A tandem repeat, right? A tandem repeat sequence, right? So these three sequences are organized in tandem and they are repeated 3 times, right? In a population, say population in Tuscarawas County, you can have the overall tandem repeat there that can be 4. In Holmes County, that tandem repeat can be 7, right? And we are going to say that the population in Tuscarawas and that of Holmes County is different in their CNVs because obviously the repeat number is different when we compare the two populations, right? And then we have gene expression. In gene expression here, we're going to focus on the pattern of genes that are overexpressed or underexpressed in a population for a particular trait, right? Now, obviously, in that condition, we have to take into account epigenetic signatures, right? Which are addition of methyl groups, okay? So this this summary slide, actually we're going to explain a little more as we go. So if there are things I forgot to underline here, we are going to line them in the coming slides. All right. In the single nucleotide polymorphism, we have sites in genomes where DNA bases are going to vary at least in 1% of the population. In the studies, the SNPs are going to span the whole genome, not one single gene, but it's going to be the whole genome, right? So we are going to take a look at the variation anywhere in the 3.2 billions of base pair that we find in the human genome. Now, whenever we do this kind of study, statistical meaning is going to be very important. When do we say that this work is statistically acceptable, right? we are going to say that the work is statistically acceptable if that work, in the case of GWAS, involves at least 100,000 markers, right? So typically, a GWAS uses millions or more SNPs grouped into a million or so per haplotype. What is a haplotype? It's basically that we are going to look at the variation on one single chromosome, right? You know that in your somatic cells, you have two genomes. One you receive from mom, one you receive from dad. And this genome, actually, for a given chromosome, you have homologous chromosomes, right? So rather than focusing on homologous chromosomes, we are going to focus on one of the chromosomes. Say the chromosome you receive from dad, right? That's what we are going to use to look at the variation, right? So we look at one single chromosome across the genome, obviously. All right, so this is an example here. So here we have one chromosome, another chromosome. So we are going to say that there is actually a single nucleotide polymorphism if we have, say, this is the sequence that happened in 98% of the population. So we have A, G, T, right? So that G pairs with C. This is in 98% of the population. That's what we see. There could be 2% of the population in which, in place of G here, we have T. In place of C here, we have A. That would be considered a single nucleotide polymorphism. How about the copy number variant? The copy number variant, again, it's a repeat, tandem repeat, right? DNA sequence that repeats, right? With a different number between individuals, right? Now, CNVs does not provide information in the same way a gene actually can encode a protein. No. But comparing CNV, it's a different way to actually compare individuals, right? To tell people apart, right? To tell people in terms of variation of the trait, right? CNV actually can be very useful in forensic science. For instance, telling who was present at the crime scene, right? Or who is innocent, right? Because we know that many people are being thrown in jail while they never committed a crime, right? So using CNV can also be used to prove that someone who has been in jail and has been saying that I'm innocent, I was not there, I didn't commit the crime, and indeed didn't commit the crime. Okay, so here is an example of CNV. So we have the DNA sequence GATTACA for allele 1 in one individual. We can have that sequence repeated twice. And in that case, we're going to have allele 2. It can be repeated 3 times. We're going to have allele 3, or 4 times allele 4. So the number of times we have the repeat is going to determine the allele and the difference between individuals. All right. Then we have another approach which is tracking genes in a group. So here we are going to isolate two groups, right? A group of people with the disorder and a group of people without a disorder. We are going to isolate their DNA, right? And then we are going to compare their DNA and look for differences, right? Which can allow us to reach a conclusion to say that, oh, these individuals here are expressing this disorder because overall we see this variation compared to individuals who don't have the disorder. Now, the GWAS is going to use a multiple step approach. So, for instance, in the initial study, we are going to have 800 people that are affected and 800 people that are not affected. We are going to look at 500,000 component of the genome and look for SNPs among this 500,000 components of the genome. Then that's the first study. In the second study, the follow-up study, we are going to narrow things down. But while we are narrowing things down, we are going to take a look at 2,000 cases of people affected and 2,000 cases of people not affected. But this time, we are not going to focus on 500,000 tag of SNPs. We are going to narrow things down to about 25,000. Okay? And then in the next study, we are still going to have a high number of affected to unaffected. However, the number of genes the number of sequences we are going to look for and SNPs, we're going to reduce it to about 100, 150, 1,500 SNPs. And then we narrow things down even much lower to the genetic level. So we're going to take a look at about 25 specific genes to see if those 25 specific genes have something to see with the difference that we see between healthy and diseased people. So GWAS study design can be done in a cohort study. In a cohort study, we are going to focus on a group of people that live in a particular area with similar environmental factors. We're going to try to identify if there is something that is specific that explains a particular trait. There was a study that was done in 1949, and that study was done in an area called Framingham. And in that area in the United States, the study was about the occurrence of cardiovascular diseases. So looking at cardiovascular diseases in the community of Framingham, researchers were interested in identifying genes that could be involved in occurrence of cardiovascular diseases among the people in Framingham, and also looked at environmental factors that would be involved in occurrence of cardiovascular diseases among these people. So this is a very well-known study in genetics. Another approach is the case control study. In here, we are pairing individuals that express a certain disease and individuals that do not express that disease, right? So here, the SNPs are going to focus on the differences in individual that have a feature compared to individuals that don't have that feature, right? So we're going to take a look at difference in SNPs. Then another approach is to look at affected sibling pair strategy. So here, that strategy is going to scan genomes of siblings for SNPs shared by those with a condition compared to those who do not have that condition, right? And then we have homozygoty mapping. In homozygoty mapping, this is going to focus more on cases of consanguinity. Okay, so when people are closely related and expressing a particular trait, so we are going to take a look at, do they share some particular genetic difference that explains why there is occurrence of that trait in that family. So this slide, it's basically to repeat the same thing. It's a summary slide. Okay, so we have a cohort. What is a cohort? Case control, what is it? Affected sibling pair, what is it? And homozygoty, mapping. Okay, what is it? So that's basically repeats same thing as what I said earlier. Now there are some limitation to genome-wide association study. What are some of these limitations? You can include so many data points. Okay, you can include so many data points, and so many people are prone to error. The GWAS also reveals association between two types of information, but it doesn't give the cause of why this feature is being observed. There are biases that can be introduced in the way the patients are selected. And that's why really the study on particular groups of people is problematic when it comes to generalizing the conclusion to humankind. The accuracy is affected by complicating factors. Those complicating factors can be, for instance, phenocopy. And phenocopy, just as a reminder, it's basically when we see a feature that appears in a population that may look like it's inherited feature, while it's actually not inherited. It's actually because of some circumstances that are not associated with genetic makeup, right? That's the case of phenocopy. And also epistasy, it's another situation that can complicate the study in GWAS. And also, we may miss some SNPs that are extremely rare in a population. Now, this is a good example of limitations of the GWAS. So here we have a child with actually blonde hair, right? So the The child here with blonde hair is a child from the Solomon Island. Okay, so the in the Solomon Island, many people actually have blonde hair. And the question at some point was, why are some people in Solomon Island have blonde hair? A study was performed and a conclusion was reached that there was a particular gene that is called TYRP1 is actually a gene that shows a particular variation in the individual with blonde hair compared to individual without blonde hair, right? So the success of the GWAS is actually based on the type of question one is asking. The more your question is well framed, the better the quality of the result you are going to achieve applying the GWAS technique, right? That that is demonstrated by the question that was asked on the Solomon Island. Okay, Solomon Islanders, the question was very straightforward. Why some of these people have blonde hair, right? And just looking at now that specific aspect, right? With considering the environment, it was possible to isolate the specific gene TYRP1. So, what are some terms used in evaluating multifactorial trait? Now, we looked at the coefficient of relatedness. Please review that. We looked at concordance. Review that. Empiric risk. Genome-wide association study and heritability. Please review the slide. Now, how about body weight? Body weight, it's a complex trait. It's a complex trait that was studied by many researchers and still today is being studied, right? Body weight can be explained by genetic makeup. It can be explained by lifestyle. It can be explained by the environment, right? So studying body weight has allowed scientists to develop what we call the body mass index. That body mass index, it's actually a measure, right? It's the weight in kilogram over the height in meter square. Okay, so this BMI actually allows to basically characterize people in relation to body weight, right? The irritability for BMI can range in a various studies, basically from 0.4 to 0.7, right? We saw that in previous slide. So as that heritability varies between 0.4 and 0.7, this actually leaves room for environmental influences, right? And our lifestyle, okay? So here we do have a chart of body mass index, and knowing your height and knowing your weight, you can actually tell your body mass index and show if you are overweight, if you are healthy, or if you are obese. For instance, me, I am 5'11". Okay? I'm 5'11". There is no 5'11" here, but there is 5'10", and there is 6 foot right here, right? Then I am about 195, so I would fit somewhere here, right? So then what I would do is to come down like this, okay, and cross this two, right? And see that I fall somewhere here and here, meaning that I am overweight, which, that's basically what the chart says, right? Just to give an example. All right, how do genes affect weight? Now, there are a number of genetic factors. For instance, leptin. Leptin, it's a hormone, right? It's a hormone that is a protein. That hormone is produced by fat cells. This hormone is going to have a number of effects on us. It's going to go in the brain and cause a decrease of appetite. Therefore, leptin is going to act in maintaining weight, healthy kind of weight. So by decreasing appetite, you are eating less and you are basically working against being overweight, right? The ghrelin, which is a peptide hormone, a peptide, it's a number of amino acids attached together via peptide bonds, right? Forming a peptide molecule, right? But this is a hormone, bottom line. This is a hormone that does the opposite that leptin does. Ghrelin is going to be produced by the stomach, and this one increases appetite, okay? And it functions as a short term to regulate weight, right? So ghrelin increases appetite while leptin decreases appetite. There are many more genes that are involved in gene regulation, in weight regulation. So we cannot attribute control of the weight based on how much leptin gene is being expressed, how much ghrelin gene is being expressed. There are many other aspects there. So here it's basically a genetic control of body weight. You are going to see that there are many genetic components. We see leptin transporters, leptin receptors. In other words, someone can produce leptin, all right, that is going to decrease appetite, but that someone who is producing leptin may have transporters of leptin that are problematic. If that is the case, you understand that even though the person produces a normal leptin, that leptin that is not carried to the destination is not going to be able to decrease appetite, which is going to cause someone to eat more, right? Leptin receptors. You can have leptin all day long. If the receptors are absent, that is a problem, right? Which means that even though fat cells are releasing leptin to say, don't eat, no appetite, well, guess what? Leptin is not reaching the destination. Therefore, the individual is not responding to leptin. Okay? So, I'm just trying to get you to understand that there are many aspects that are involved in body weight, right? So please review some of this. I'm not going to specifically ask you to master all of this. I want you to understand the concept, which is that many genes are involved in the control of body weight. That's one. Two, because many genes are involved in the control of body weight, it's not obvious to just come up with one particular gene that is responsible for someone being overweight, right? You have to take into account all these genes. Now, also, you have to understand that all the genes that are listed here are being involved in the control of body weight. It's for what we have discovered. Question: are there some genes that we have not discovered to be involved in the control of body weight? It is possible. All right? It is possible. So things are more complicated than just knowing simply the genes. All right. The environmental influence on body weight. So obviously, what we eat, how much we eat, is going to be very important, right? The climate, believe it or not, is going to affect our body weight. But a very important piece I want to bring here, it's actually the role of the microbiome in your gut. Your gut microbiome are going to play a very important role in your body weight. This has been shown. And that is the end of this chapter. Thank you for your attention, and I will talk to you again another time.